Okay, here we go. Let's talk a little bit about protein synthesis. Protein is one of the most important molecules in your body. They handle chemical reactions, they make up your muscles, your ligaments, your tendons, everything that helps keep you alive. Without proteins, you will quickly run out of ways to make chemical reactions happen in your body. So, since they're so important, we keep the information stored away in the nucleus of each of your cells. So, the safest place in every possible cell has DNA in it because it's the code for proteins. That's how important proteins are. So, the major idea I want you to get from this is that DNA is used to make RNA, and RNA is used to make proteins. So if you look at a DNA molecule, it's divided up into several genes. The genes basically are coding regions that actually code for a specific protein. You've got one there, you've got another one here, and you've got a third one here. If you zoom in on one segment of this gene, you'll see that it's a nucleotide sequence, just like any other piece of DNA. This nucleotide sequence is going to be copied because it can't leave the nucleus into something called RNA, or messenger RNA. Messenger RNA is divided up into 3 nucleotide pieces called codons. That thing's called a codon. Each codon codes for a specific amino acid, and you'll see how that works in the chart we're going to look at next. So, right here, I had a DNA nucleotide sequence of AAA, and that's going to be transcribed into an RNA sequence of UUU, which is complementary to it. If I look at this chart, notice that codon UUU. If I find UUU right up here, it's phenylalanine. Phenylalanine is a specific amino acid. So when I go back to the thing I was just looking at, this square right here represents phenylalanine. This is going to represent another one. So you get a long chain of amino acids eventually because of this code that is found in DNA and copied to RNA. So if you remember, amino acids are what makes up proteins. I don't know why I went past that. When you look at this genetic code, there's also a couple highlighted categories. We have this green one right here, which is methionine, which is the start codon. So that tells the protein to start right there. So all proteins will start with methionine. We call it the start codon. You also have several stop codons. The stop codons will tell the RNA to stop transcribing. That's the end of the gene. So you go from start to stop, and you get a protein at the end. Here's the way it works. Inside the nucleus, you have DNA. DNA is too big to get out of the nucleus, so it needs to make a copy. This copy is made in messenger RNA. This messenger RNA will take that message out of the nucleus because it does fit through the nuclear pores and go out to the ribosomes. The ribosomes are the little protein factories. This is telling the ribosome what to make. So this is the instructions. The messenger RNA is the instructions. And once the ribosome reads it, brings in the amino acids, puts them in a long chain just like this, you get a polypeptide. When that polypeptide is folded properly, it is now a protein. So a recap of that process. First you make the DNA into messenger RNA. That process is called transcription. So transcription is DNA to RNA. Then, once the messenger RNA leaves, it's translated by the ribosome and transfer RNA into this amino acid sequence, which makes up a protein. So, there it is. The way to think about this is DNA is too important to leave. So it stays in the nucleus, and it's kind of like a file stored on your computer. In order to make a copy you can carry around, you make a little thumb drive copy of that one file, not an entire hard drive. And RNA is kind of like your thumb drive copy that you can take places. Hope that makes sense. If not, whatever, I'm moving on. Here's what RNA is. RNA is a nucleic acid, just like DNA, so it's made of repeating nucleotides. RNA is usually a single strand. It's not normally double-stranded, though it does exist sometimes. There's no thymine in RNA. It's replaced with a pyrimidine base called uracil. So you'll see U pairing with A. Also, there's the matter of the sugar that it has. It's got ribose instead of deoxyribose. That's why it's called ribonucleic acid. So the 3 forms are right here. Messenger RNA is the first one over here on the left. You'll see it's a single strand. Transfer RNA is the second one. Transfer RNA carries amino acids with it. And it has something at the bottom called an anticodon. If you remember, messenger RNA has something called codons. And every 3 nucleotides, let me turn my little pen on real quick. So let's pick, let's take these 3 nucleotides right here. There's 1, 2, and 3. So those 3 nucleotides are a codon. I'll click them all together. So if you can imagine, if this anticodon matched up with this one, this would move in and plug itself in right here, and then you have this amino acid ready to go in the right place. Then another transfer RNA would come in for these 3 codons right here, and its anticodon would bring in another amino acid, and that amino acid would be right here. I'll make that into a little square-ish looking thing. And eventually when they're together, they'll join together with a peptide bond, and you're on the way to making a protein. This over here, let me get rid of that. This over here is ribosomal RNA. This is what ribosomes are made of. They're made of this and some other proteins that are manufactured, but their structure is not as important. But I'm telling you right now that ribosomal RNA makes up ribosomes. Makes sense. Here's messenger RNA. Messenger RNA is a single coiled chain of nucleotides, and it is a copy of a gene that's found in DNA. So it carries the information from the nucleus out to the ribosomes for proteins to be made. This is important, the codons part. Its nucleotides are in little groups of 3. Every group of 3 is called a codon, and every one of those is a code for a specific amino acid that belongs in that order. Here's messenger RNA. Messenger RNA also, to protect itself from accidentally being digested or getting its message erased, it's got a protective cap and tail at either end. The actual message doesn't start until somewhere in the middle, right here. So there's the start of the actual message, and here's the end. So the start codon will be right here, and that should be A, U, G. And then one of those stop codons will be at the end, so maybe something like U, A, oh, this looks terrible, A. So that says UAA if you can't read it. Okay, so that's a stop codon, and this over here is a start codon. Transfer RNA is a little different because its nucleotide sequence does not matter. So all of this stuff that's in here, all these A's and U's and T's and C's, that doesn't matter. The only part that does matter is the part down here called the anticodon. The anticodon is complementary to a codon on a messenger RNA molecule. So they pair up together. When they pair up, this amino acid, whatever it is, will be brought to the right place. And remember, this is all happening at the ribosome, so you'll see what that looks like later. So here's kind of what it would look like. This is what an actual RNA molecule is kind of looking like. This is a representation that's a little bit confusing, so instead we're going to turn this into this over here. Hopefully you can understand because it's been color-coded. The blue part is where the amino acid goes. This green part here, all these complicated nucleotides, again, it doesn't matter which nucleotides they are. They're going to become this green cylinder here. And at the bottom is the anticodon. I told you that was important, and that is going to end up over here. So here's the representation of a tRNA molecule, transfer RNA, that we're going to use. At the top, there's the amino acid being carried. Here's ribosomal RNA. This is what ribosomes are made of. It's arranged in globular form, and that means what it sounds like, which means it looks like a glob. So this is RNA and proteins. And here's a look at a ribosome. So they look like this. There's a small subunit and a large subunit, and they're divided by a space right here where the messenger RNA runs through and gets read like a printer. So you run it through there, it rolls its way through, and as it moves through, transfer RNAs bring in the appropriate amino acids and they make a big chain. This may look confusing now, but just so you understand, this is going to ride through the ribosome like rollers. If that makes sense. I don't know if it does. If not, whatever. And the transfer RNAs come in as the messenger RNA goes by, and they attach their anticodons to the messenger RNA codons, and they bring in each amino acid that needs to be there. So answer these questions real quick. So go ahead and pause it, and I'll wait. Okay, so what is mRNA for? Messenger RNA is for bringing the message. How am I trying to say this? Messenger RNA is for bringing a copy of the DNA out of the nucleus and to the ribosome. Transfer RNA is a little bit different. Transfer RNA is meant to bring an amino acid in and put it in the right place. How are they different? The major way that they're different is that messenger RNA's nucleotide sequence does matter, and the transfer RNA's sequence does not matter, except for the anticodon. Our RNA, that's what makes up the ribosome. Here's my little overview of protein synthesis. Proteins are important because they control genetic traits, and they also control every chemical reaction in your body by virtue of being enzymes. All enzymes are proteins. So, two things have to happen before those proteins are actually manufactured. The first is transcription, and that's where DNA is transcribed to messenger RNA. And the second is translation, and that's where the messenger RNA codons get translated by transfer RNAs at the ribosome into an amino acid sequence that will turn into a protein. Here's what transcription is. DNA is going to be transcribed into RNA by an enzyme called RNA polymerase. Very similar to DNA polymerase, only it brings in RNA nucleotides instead. So RNA polymerase is going to start at a piece of DNA that we call the promoter, and that's going to start the gene. So the gene begins at a place called the promoter, which is going to contain the start codon. It will all stop at the termination signal or terminator, which is where the stop codon is. And that tells RNA polymerase, you can stop, you can get off right here. Here's what that means. So the blue is the DNA molecule. RNA polymerase is a big old molecule. It's going to attach to a point. And that point on the DNA molecule is where it's going to start. It's called the promoter. So the RNA polymerase binds to a promoter and starts putting in RNA nucleotides, just like this, complementary base pairing, just like DNA replication. So it's going to be transcribing this RNA molecule. So this is getting made right here. So here's the steps of transcription. There's the RNA polymerase molecule. It's going to bind to the promoter and start moving along in this direction until it reaches the terminator. So you can see it moving along here, and as it moves, you can see this RNA molecule getting longer and longer. So it's going to move, and when it finally gets to the end, when it gets to the terminator signal right here, it's going to stop. It's going to cut it off. So you have a complete messenger RNA molecule right here as a result. So that's how the gene gets transcribed, and that is going to leave the nucleus. Okay, now the next step is translation. Translation is called translation because the nucleic acid language that it's being used is going to be translated into an amino acid sequence. So you've got a bunch of A's, T's, C's, and G's, and you need to turn that into specific amino acids. Transfer RNA is going to do that, and the place it does it is at the ribosomes. If you even know what tape in a VCR is, you can think about it like this. You remember those old movies that your grandma has, and there's tape in a little plastic box, you put it in a VCR and it reads the tape through these little rollers. That's how the ribosome works, basically. So the transfer RNA is brought into the ribosome and it has an anticodon. It's going to recognize a certain codon at a certain place on messenger RNA and bring in the appropriate amino acid. And those amino acids, when they're next to each other, are going to bind to each other and make a long chain. So it starts at the start codon, which is always AUG, and it's going to end at a stop codon. Okay, here's what it looks like again, just a refresher. Here's how it works. Kind of complicated. You've got two little sites. You've got a primary site and an auxiliary site. So the first tRNA is going to land right there. Then another one is going to land right next to it, at the A site. One of them is going to hold a long chain of amino acids, and it's going to pass that off to the next one, and they will move through. So each RNA is going to move through. So it always is going to start with methionine. So there it is, number one. First one starts right there. You can see it binds in, and it's going to be at the A site, or excuse me, at the P site. The next steps are called codon recognition, peptide bond formation, and translocation. So you can pause it and write those down if you need to. And since I don't have a whole lot of time, I'm going to move on to the next part. We will talk about this a little bit more in class because this is the most complicated part, but this is how the chain gets lengthened. So you can pause and take a look at this. And this gives you the whole process. So take a look at this, take your notes, and get